The overall goal of the following experiment is to energize recipient allospecific donor T cells by ex vivo stimulation with recipient alloantigens in the presence of antibodies that block antigen-presenting cell co-stimulation of T cells via their CD28 receptor. This is accomplished by first isolating PBMC from two different healthy donors. Next, a bulk alloenergizing co-culture is set up by incubating the HLA-mismatched PBMC populations in the presence of monoclonal antibodies to the ligands B7.1 and B7.2 to block CD28 stimulation. Then, the efficacy of the alloenergization is measured by primary and secondary mixed lymphocyte reactions, or MLR. Finally, the allospecificity of the alloenergization is measured by determining third-party and CMV-specific responses. Results are obtained that show that donor T cells are rendered hyporesponsive to subsequent alloantigenic stimulation, but that non-alloreactive donor T cells are left intact and functional.
We've successfully employed this strategy in previous and ongoing pilot clinical trials of infusion of alloenergized donor T cells during and after HLA mismatched stem cell transplantation and observed improved immune reconstitution, fewer infections, and less severe acute and chronic graft versus host disease than seen in historical control recipients of unmanipulated HLA mismatched transplants.
A major advantage of the strategy of alloenergization is that off-target effects are minimized, as the technique does not reduce pathogen-specific or tumour-associated antigen-specific T cell responses or eliminate CD4 regulatory T cells from the donor T cell pool. Demonstrating the procedure will be Christine Barban, a staff scientist in our laboratory.
The first step is to draw blood from two healthy volunteer donors. Cells from one donor will serve as the responder, and the cells from another donor will serve as the stimulator. Next, isolate PBMC from the blood by density gradient centrifugation using Ficol HiPAC. Stain a small amount of cells with trypan blue and count viable PBMC using the hemocytometer. Resuspend PBMC from each donor in 10 ml complete culture media at a concentration of 1 million viable cells per milliliter in separate 15 ml conical tubes, designating one tube of donor PBMC as stimulators, the other as responders. To set up the bulk alloenergizing co-culture, add 100 micrograms of anti-B7.1 and anti-B7.2 antibodies to the 15 ml tube designated stimulator. Gently agitate the tube and incubate for 30 minutes. After the incubation period, irradiate the stimulator PBMC and add them to a T25 50 cubic centimeter cell culture flask with a gas permeable cap. Label the flask bulk alloenergizing co-culture. Next, add 10 ml of responder PBMC from the other donor at 1 million per milliliter in complete media to the flask. Add a further 100 micrograms of each of the anti-B7.1 and anti-B7.2 antibodies to the flask and mix gently prior to placing the flask upright in an incubator for 72 hours. Next, irradiate another 10 million of the stimulator PBMC for a bulk control co-culture without blocking antibodies, and then add them to a T25 50 cubic centimeter cell culture flask. Label the flask bulk control co-culture. Add 10 ml of the same responder PBMC as used for the bulk alloenergizing co-culture at 1 million per milliliter in complete media to the flask and mix gently prior to placing the flask upright in an incubator for 72 hours. The next step is to measure the function of the alloenergized donor T cell pool. On the same day as the bulk co-cultures, label one U-bottom 96-well plate primary MLR for each of the 3 time points to be measured. Then, decant 5 ml from each of the bulk control and alloenergizing co-cultures into 15 ml tubes. Next, pipette 200 microliters of cell suspension from each tube into triplicate wells on each of the 3 plates. Label these wells no co-stimulatory blockade or no CSB for the cells from the bulk control co-cultures and CSB for the cells from the bulk alloenergizing co-cultures. Then, add 200 microliters of complete media to 6 wells on each plate as negative controls. Finally, add 200 microliters of PBS to all the empty wells to reduce evaporation and place the plates in the incubator. 72 hours after setting up the bulk co-cultures, label one U-bottom 96-well plate, secondary MLR, for each of the 3 time points to be measured. To prepare the responder cells for the secondary MLR, Transfer 5 milliliters from each of the bulk control and alloanergizing co-cultures to 15 milliliter tubes and centrifuge the tubes for 5 to 10 minutes at 2000 rpm at room temperature. Then, wash the cells 2 more times by carefully aspirating the supernatants, disrupting the pellets, adding 10 milliliters of complete media to each tube, and then centrifuging the cells again. After the second wash, resuspend the cells in 1 milliliter of complete media. Use trypan blue staining to enumerate the viable cells and adjust the cell concentration to 1 million viable responder cells per milliliter. Pipette 100 microliters of cell suspension from each tube into triplicate wells of each plate. Label these wells First Party or FP allo-restimulation. Then pipette 100 microliters of cell suspension from each tube into another 3 wells on each plate and label these wells CD3/28 stimulation. PBMC from the same donor used to supply first-party stimulator PBMC for the alloanergized and control co-cultures are used for stimulator cells for secondary MLR. Suspend 10 million of the first-party donor PBMC at a concentration of 1 million per milliliter and irradiate. Add 100 microliters of irradiated stimulator cells to the wells labeled FP allo-restimulation on each secondary MLR plate. For positive controls, add 1 microliter each of CD3 and CD28 monoclonal antibodies and 98 microliters of complete media to the wells labeled CD3/28 stimulation on each secondary MLR plate. In addition, add 200 microliters of complete media to 6 empty wells on each plate to serve as negative controls, and add 200 microliters of PBS to all empty wells to reduce evaporation. Place the plates in the incubator. The next step is to measure the specificity of the alloanergization. PBMC isolated from a new healthy donor, termed the third-party donor, are used to test the specificity of the alloanergization process in a secondary MLR. Suspend the third-party PBMC at 1 million cells per milliliter and irradiate. Label 3 96-well plates secondary MLR specificity day 3, 5, and 7. To prepare the responder cells for the secondary MLR to measure specificity, transfer 5 milliliters from each of the bulk control and alloanergizing co-cultures to 15 milliliter tubes and centrifuge the tubes for 5 to 10 minutes at 2000 rpm. Then, wash the cells twice by carefully aspirating the supernatants, disrupting the pellets, adding 10 milliliters of complete media to each tube, and centrifuging the cells again. After the second wash, resuspend the cells in 1 milliliter of complete media. Use trypan blue staining to count the cells and adjust the cell concentration to 1 million viable responder cells per milliliter, and then pipette 100 microliters of cell suspension from each tube into triplicate wells on each plate. Label these wells TP or third-party allostimulation. Then add 100 microliters of irradiated TP stimulator cells to the wells labeled TP allostimulation. To further test the specificity of alloanergization, responses to pathogens are measured after energization of PBMC from donors with previously established proliferative responses to CMV. Label 3 96-well plates secondary MLR CMV day 3, 5, and 7 and transfer 5 milliliters from each of the bulk control and alloanergizing co-cultures to 15 milliliter tubes. Then wash, count, and resuspend cells at 1 million per milliliter in complete media. Pipette 100 microliters of cell suspension from each tube into 3 wells on each plate and label these wells CMV stimulation. Then add 0.1 microliters of CMV lysate in 100 microliters complete media to wells labeled CMV stimulation. Then add 200 microliters of complete media to 6 wells on each plate as negative controls. Finally, add 200 microliters of PBS to all empty wells to reduce evaporation and place the plates in the incubator. To measure proliferation in primary and secondary MLR, add 1 microcurie of tritiated thymidine to the negative control and cell-containing wells 16 hours prior to harvesting the plates. Collect the thymidine on 1 filter mat for each plate using a Tomtec harvester or similar. Air dry the filter mats for 1 hour, then place them in individual sample bags. Add 5 ml scintillation fluid to each and seal. Measure the tritiated thymidine incorporation in the last 16 hours of the experiment by reading the filter mats with a Wallach microbeta scintillation counter or similar. Using HLA mismatch stimulator and responder PBMC, it can be seen in this figure that the presence of co-stimulatory blockade in the primary MLR reduces mean alloproliferation of responder PBMC at day 5 to around 30% of that seen in control primary MLR in the absence of co-stimulatory blockade. This is equivalent to a mean inhibition of primary alloproliferation of around 70% at day 5, as seen in this figure. It can be seen in this figure that in the secondary MLR, first-party alloproliferation of alloanergized PBMC at day 5 is typically 10 to 15% of that seen with control PBMC. This is equivalent to a mean inhibition of proliferation of 85 to 90%, demonstrating that alloanergized PBMC are hyporesponsive to first-party allostimulators, as seen in this figure.
Once mastered, bulk co-cultures to alloanergized donor peripheral blood mononuclear cells can be set up in 2 to 3 hours. The approach can be easily standardized for clinical use to generate donor T cells with reduced alloreactivity but retained pathogen-specific immunity. Such cells can be adoptively transferred in the setting of allogeneic hematopoietic stem cell transplantation to improve immune reconstitution without excessive graft-versus-host disease.
While attempting this procedure, it's important to remember to work quickly. This helps preserve the viability of cells and avoid nonspecific activation or loss of antigen-presenting cells. If using cryopreserved PBMC for stimulators or responders, a validated protocol for resuscitation of frozen cells that gives consistently high yields of viable cells should be used.
After watching this video, you should have a good understanding of how to alloanergize human donor peripheral blood mononuclear cells, and how to perform assays to assess the efficacy and specificity of this procedure. The strategy can be used to generate human donor T cells with reduced allo responses, which can be used clinically to improve immune reconstitution after allogeneic hematopoietic stem cell transplantation.